Chemically, hormones are of different types. They can be proteins, polypeptides, they can be steroids, or they can be amines. However, fundamentally, they act by the same mechanism by binding to their receptor. And this receptor can be present on the membrane, which are cell membrane receptors, or they can be present intracellularly. And when intracellularly, they can be of two types. Either they can be within the cytoplasm, so that is cytoplasmic receptor, or they can be present within the nucleus, so that is nuclear receptor. So fundamentally, this hormone binds to the receptor wherever the receptor is present and forms the hormone receptor complex, which in turn leads to the activation of downstream events and hence leading to changes in the cell function. For example, the action of gastrin on parietal cells leads to secretion of the hydrochloric acid. On the other hand, the action of other hormones, say suppose the action of somatostatin on gastrin, leads to decrease in the release of the gastrin. So it can be both ways. It can lead to increase in the function, or it can lead to decrease in the function, or it can lead to change in the function also, right? So how this is brought about, that we will see one fundamental scheme, and then we will move to individual mechanism of action. So with the formation of the hormone receptor complex, there are cascade of events which are started. So here it is showing this is the receptor and this is the hormone which has bound to its receptor. So with this, there is activation of the cascade of events which basically are of two types: increase or decrease in the activity of enzymes, or increase or decrease of gene transcription, leading to the either more synthesis of proteins or decrease in the synthesis of proteins. Increase or decrease in enzyme activity is brought about mainly by phosphorylation. Phosphorylation is attachment of a phosphate group to an protein or an enzyme. Okay, so here you see that there are two types of enzymes which are activated, that is kinases and phosphatases. With the formation of the hormone receptor complex, first there is activation of a kinase, and kinase causes phosphorylation of the protein. So here we are seeing that there is a protein. With the activation of a kinase, there is addition of a phosphate group to the protein. And with time what happens, that this phosphate group is removed also. So there are phosphatases also which are activated in some time difference. That is, there is a little bit delay with the activation of the kinase, but they are also activated, and this leads to removal of the phosphate group from the protein. So that yes, the action is brought about, but after some time the action is terminated also. So this concept is known as phosphate timer, that is activation of the kinase and then activation of the phosphatases, so that the function is brought about and after some time it is stopped also. Then moving on to the other fundamental mechanism, that is increase or decrease in gene transcription, that is brought about by the activation of the transcription factors. And these transcription factors, either they are present in the nucleus itself, okay, so there is activation of MAP kinase. Because of this formation of the hormone receptor complex, there is activation of MAP kinase which moves to the nucleus and phosphorylates the transcription factor which is present in the nucleus. So that is one mechanism. Second is that the transcription factor might be present in the cytoplasm, but it is inactive. So there is activation of this transcription factor which is present in the cytoplasm by the phosphorylation of this transcription factor. Okay, so the one example is STAT, that is signal transducer and activator of transcription. So we will see the mechanism later. Then third mechanism is that the hormone receptor complex itself moves to the nucleus and binds to the certain regions in the DNA known as hormone receptor elements. And this leads to increase or decrease in the synthesis of proteins. So these are three mechanisms by which there can be increase or decrease in the gene transcription. Presence of the transcription factor within the nucleus only, that is phosphorylated, or presence of the transcription factor in the cytoplasm which is phosphorylated. So by phosphorylation these are becoming active, or there is presence of hormone receptor complex which itself binds to hormone responsive elements within the DNA, right? Now, most of these mechanisms, this increase or decrease in the enzyme activity and these activation of the transcription factors, this is brought about mainly by the cell membrane receptors. Okay. So cell membrane receptors, the ligands, when bind to cell membrane receptors, these different kind of mechanisms can be activated. However, this formation of the hormone receptor complex and increase or decrease in gene transcription is brought about by binding of the ligand to the receptor which is present within the cytoplasm or in the nucleus. So now moving on to individual mechanisms. So we have different types of receptors. As I already told you, there are cell membrane receptors and mainly the hormones which are of protein, polypeptides and the catecholamines, that is epinephrine and norepinephrine, bind to these cell membrane receptors. Then we have the cytoplasmic receptors. So steroids, mainly the steroids have the cytoplasmic receptors, so they act by binding to them. And then we have the nuclear receptors, and nuclear receptors mainly the thyroid hormones, vitamin D and vitamin A. These are the ones which act by binding to the nuclear receptors. So what are the mechanisms by which these act? So there are different types of mechanisms. First we will discuss the cell membrane receptors, and these cell membrane receptors again are of three different types. We have the ion channel linked receptors, G protein coupled receptors, that is GPCR, or G protein linked receptors, also known as G protein linked receptors, and then we have enzyme linked receptors. So as you see the name, that these receptors are linked or coupled. Okay, so we have a receptor, so and it is a cell membrane receptor. So say suppose this is the cell membrane and here this is the receptor. So these receptors are linked either with ion channel, or they are linked with G protein, or they are linked with an enzyme, right? So first let us see what is this ion channel linked receptor. This ion channel linked receptor is also known as ligand gated channel, or it is also known as ionotropic receptor. Okay, so what is this ion channel linked receptor? See here, this is the membrane, and outer side this is the extracellular side, and this is the cytoplasmic side. So this is the receptor which is shown, and there is the binding site for the ligand. So ligand, when it comes, it binds to this receptor. Actually this receptor is also a channel. So with the binding of the ligand, there will be opening of these channels. Okay, so these are known as ion channel linked receptors. When the ligand binds, there will be opening of these channels, and mostly the neurotransmitters act by this mechanism. So we have mainly the acetylcholine, glutamate, GABA. They act by this mechanism, that is by binding to ion channel linked receptors. So this is very simple. The ligand binds and there is opening of the channel. Moving on to the next mechanism, that is the G protein coupled receptor. Now this G protein coupled receptor, as the term indicates, that it is linked to G proteins. Okay, so this is the membrane, and here this diagram is showing a G protein coupled receptor, and as you see that this receptor has the structure such that it crosses the membrane 7 times. Okay, so if you count the number of times it is crossing the membrane, it will be 1, 2, like this, 3, 4, 5, 6, 7. So 7 times it is crossing the membrane. So that is what we call it. It is having 7 transmembrane domains. 7 transmembrane domains. Okay, that is why it is also known as 7 helix receptor or serpentine receptor. Serpentine receptor. Okay. Now outside towards the cytoplasm, it has a site where a ligand can bind, and inside there is a site where heterotrimeric G proteins can bind. Heterotrimeric G proteins, meaning what? Heterotrimeric means that there is this G protein which is having 3 different units, and these units are G alpha, G beta and G gamma. So this receptor can bind to these 3 units. Okay. Now, what happens that when the ligand binds, there is some changes in these units such that there is an activation of nearby enzyme. Either there can be activation or there can be inhibition. So based on that, whether there is activation of the enzyme or inhibition of the enzyme or which enzyme is being activated, we have 3 different types of GPCRs also. Okay, so we have GS kind of heterotrimeric proteins, that is stimulating adenyl cyclase enzyme. Okay, so this enzyme which is drawn here, it can be adenyl cyclase, and GPCRs can stimulate adenyl cyclase. I for they can inhibit adenyl cyclase. Okay, and GQ is the GPCRs which stimulate the phospholipase C enzyme. Phospholipase C enzyme. Okay, so remember GPCRs, GS, GI, GQ, and they all act by different mechanisms, but fundamentally there will be phosphorylation of the proteins. Okay, so we will discuss each of them one by one. So first is the adenyl cyclase CMP second messenger system, GPCR, which activates adenyl cyclase CMP second messenger system. Now, why I have used the term second messenger? See, the first messenger is the ligand, the extracellular ligand which is binding to the receptor. But that hormone itself, that ligand itself, it is not bringing out any further changes. It just binds to the receptor, leads to the activation of the receptor, and further the changes are brought about by other chemicals which are formed. Okay, so that is known as second messengers which are carrying out the events intracellularly. Okay, and these second messengers are of different types. So we will see in GPCR there are different types of second messengers. So one is adenyl cyclase CMP second messenger. So here you see again, this is the membrane and this is the extracellular side and this is the cytoplasmic side, and we have shown the serpentine receptor. Now nearby here there is heterotrimeric G proteins with the G alpha, G beta and G gamma subunit. Now this G alpha is bound to GDP, that is why the term G. Okay, so it binds to GDP, that is guanyl diphosphate. When the ligand binds to the receptor, two things happen. One, these G proteins associate with the receptor, and because of this association, now GDP is replaced by GTP. So G alpha now binds to GTP. Then because of GTP binding, this G alpha dissociates from G beta and gamma. Okay, so G beta and gamma remain bind here and they also bring about certain functions. However, G alpha moves away with the GTP. Okay, so GTP is bound to G alpha, it moves away from its two components and then it goes and binds to an enzyme adenyl cyclase nearby on the membrane and leads to its activation. Now, this activated adenyl cyclase enzyme converts ATP to CAMP. So, CAMP is formed and this CAMP brings about multiple actions. One, it can open or close other ion channels which are present on the membrane. Okay, so here also it is different from that of the ion channel linked receptors. It is indirect opening of the ion channels because of the formation of CAMP. Then second, it leads to the activation of protein kinase A. Okay, you remember CAMP. Okay, so A for here protein kinase A. So inactive protein kinase A, which is CAMP dependent, so it is known as CAMP dependent protein kinase A. It is activated by CAMP to active protein kinase A, and as the name indicates, it's a kinase. So kinase will lead to phosphorylation. So that is the second thing it brings about, that protein kinase A phosphorylates certain proteins or enzymes, because of which there will be certain downstream events. Okay. Third mechanism of action is that this active protein kinase A can also move to nucleus and activate the transcription factors. So this is CAMP regulated element binding protein. Just remember CREB here, which is phosphorylated within the nucleus, and it also leads to increase or decrease in the synthesis of certain proteins. Okay, so you see the varied mechanism by which this CMP is acting. It can open the ion channels. It can cause the activation of protein kinase A, which in turn phosphorylates proteins. It can also lead to the phosphorylation of the transcription factors. So the cytoplasmic events which are happening, these are, you see, fast acting, because the other one where there is increase or decrease in the synthesis of the proteins, that will take some time. Okay. However, the other events, the proteins are already there. They just need to be activated. The enzymes need to be activated and the function will start. Okay. So there are certain fast actions and there are certain actions which take little time to act. Okay. So that is adenyl cyclase CMP second messenger system, where the adenyl cyclase is stimulated. But I told you that there are other types of GPCR also where it will inhibit the adenyl cyclase enzyme. Okay, so with the inhibition, what will happen that this adenyl cyclase, somewhat it is active, but if it is inhibited, then already present CMP, it will decrease. Okay, so less CMP will form, so there will be less activation of protein kinase A and less phosphorylation. So already what functions were active, now they are going to be decreased. Okay, so we have GS and GI second messenger systems. Some examples of hormones which act by this mechanism include luteinizing hormone, FSH, then vasopressin, especially the V2 receptors are there, catecholamines, beta receptors. They act by this adenyl cyclase CMP second messenger system. Moving on to the next mechanism, that is the phospholipase C IP3-DAG second messenger system. So this is brought about by GPCR, which is associated with GQ heterotrimeric G proteins. The mechanism is same. The ligand will bind to the receptor and there will be G alpha, G beta, gamma subunits which will associate with the receptor. G alpha, there will be replacement of GDP with GTP. G alpha will dissociate and go and activate another enzyme, in this case, which is phospholipase C. This phospholipase C breaks down a lipid in the membrane, and that lipid is phosphatidyl inositol biphosphate. Okay, phosphatidyl inositol biphosphate. Phospholipase C breaks down phosphatidyl inositol biphosphate into two chemicals, that is inositol triphosphate IP3 and diacylglycerol. Both of these act as second messenger, hence the name phospholipase C IP3-DAG second messenger system. Now this IP3 binds to IP3 receptors which are present in the endoplasmic reticulum and also in the mitochondria. Okay, IP3 receptors are present in these two organelles. IP3 binds to it and causes the release of calcium. Endoplasmic reticulum and mitochondria are storehouses for calcium. So there is a release of calcium and further there is downstream events. So this calcium here also acts as a second messenger, which we will see subsequently. Then coming to diacylglycerol. Diacylglycerol, it causes the activation of protein kinase C. Okay, in CMP we saw that a CMP dependent protein kinase A is activated. Here DAG dependent protein kinase C is activated, and it also causes the phosphorylation of enzymes. Okay, because it also is a kinase, right? So moving on to that, how calcium acts as a second messenger. So that is known as calcium colmodulin second messenger. Now this calcium, as we saw, that it is released by inositol triphosphate, and there will be increase in the concentration of calcium in the cytoplasm because of this. Normally the concentration of calcium in cytoplasm is very less. Okay, so with the release of the calcium, the concentration of calcium increases in the cytoplasm and it binds to a protein known as calmodulin. Okay, and here what I have shown, that calcium not only releases from the intracellular stores, but calcium can also come from outside. We have voltage gated calcium channels, we have ligand gated calcium channels, we have store operated calcium channels, where the calcium which is released causes the opening of these store operated calcium channels, causing more entry of calcium from outside. And we also have receptor operated calcium channels, and these receptor operated calcium channels are opened by the diacylglycerol. So diacylglycerol, we saw before that it is activating the protein kinase C. But here there is another mechanism where diacylglycerol is also causing the opening of receptor operated calcium channels. Then this calcium binds to calmodulin, and this calmodulin can bind to 4 calcium ions. 4 calcium ions. It has 4 sites where calcium can bind. And when 3 or more calcium ions bind to calmodulin, calmodulin causes the activation of calmodulin dependent kinases. So again kinases have come. This time they are calmodulin dependent kinases. And this calmodulin dependent kinases, we have one enzyme, myosin light chain kinase. This is present in smooth muscle. Okay, in a smooth muscle, calmodulin activation leads to activation of myosin light chain kinase, which in turn phosphorylates the myosin, and then only it becomes active. Then we have calmodulin dependent kinases 1 and 2, which are important for synaptic transmission. Calmodulin dependent kinase 3 is important for protein synthesis, and there is calcineurin also, which is important for T cell activation. In fact, this calcineurin is inhibited by certain immunosuppressants. Okay, immunosuppressants. So these are 5 major types of enzymes which are activated by calmodulin. So that was the mechanism of action of GPCRs, right? So what we saw till now, we have seen ion channel linked receptors, we have seen G protein coupled receptors, and in G protein coupled receptors we saw different mechanisms, how activation of adenyl cyclase takes place by GS, inhibition of adenyl cyclase, and there is activation of phospholipase C. Then in that only we saw the calcium calmodulin system as well, because IP3 acts by releasing of the calcium. So we have seen different second messenger systems also, right? Moving on to the enzyme linked receptors. What are these enzyme linked receptors? So as the name indicates that these enzyme linked receptors are linked with an enzyme. Now that can be an intrinsic enzyme activity, the receptor itself is having an enzyme activity, or there can be association of enzyme, a nearby enzyme is associated to it. That means the receptor itself does not have the activity, but nearby enzyme is associated with it, and with the binding of the ligand there is activation of the enzyme. Okay, so intrinsic enzyme activity or linked to an enzyme, and there are mainly 3 types. We have guanyl cyclase linked receptor, we have the receptor with intrinsic tyrosine kinase activity, which leads to the activation of MAP kinase pathway, MAPK, MAP kinase pathway, or there may be external tyrosine kinase which may be associated with it, and this leads to activation of the JAK-STAT pathway. So JAK is the tyrosine kinase and STAT activation occurs, so that is JAK-STAT pathway. Fourth one is also there where the enzyme is having serine threonine kinase activity. Serine threonine kinase activity. Actually, this tyrosine, serine threonine, these are the amino acids, and the enzyme will lead to phosphorylation of either the tyrosine, serine or threonine. So based on where the phosphorylation is occurring, we have that particular kinase. So tyrosine kinase will phosphorylate the tyrosine residues in the protein. Okay. So let us see one by one how these enzyme linked receptors are acting. We have guanyl cyclase linked receptor. So you see that here is the membrane, and this is the guanyl cyclase linked receptor, and if a ligand atrial natriuretic peptide acts by binding to guanyl cyclase linked receptor. So when the ligand binds to the receptor, there is activation of the guanyl cyclase and it leads to conversion of GTP to cGMP. Okay. Now this cGMP again activates protein kinase, a different protein kinase, and because of this activation there will be phosphorylation of the proteins. Okay. Then there is another type of guanyl cyclase, that is soluble guanyl cyclase. So that is just different. I am just explaining here. So here it is the receptor linked guanyl cyclase, okay, so that is attached to the membrane. There is another guanyl cyclase which is soluble, okay, so it is present in the cytoplasm, and that is activated by a gas, that is nitric oxide, okay. Same mechanism of action, that when that will be activated, it will lead to conversion of GTP to cGMP, and that will bring about further actions. Nitric oxide causes vasodilation, so with the formation of cGMP there will be vasodilation, okay. Now here I have shown phosphodiesterase also, that this cGMP when formed by means of phosphodiesterase, it will be broken down into 5-GMP. See, that we are going to see almost at the end of the video, that what is the mechanism to stop the various responses. So one of this is that phosphodiesterase breaks down the second messenger which is formed. So that is going to stop the response, because it's not that once activated the response should go on indefinitely. It has to be stopped as well, right? So this is one mechanism. Moving on to the second enzyme linked receptor, that is a receptor tyrosine kinase, okay. So this receptor has intrinsic enzyme activity, receptor tyrosine kinase. One example of the ligand which binds to receptor tyrosine kinase is insulin. Not only insulin, actually many growth factors like epidermal growth factor, platelet derived growth factor, all act by binding to receptor tyrosine kinase. So when this receptor binds with the ligand, actually this receptor has two units. So with the binding of the receptor, there is dimerization of the receptor. So its two units come closer, known as the dimerization of the receptor, and this leads to partial activation of the tyrosine kinase enzyme activity of the receptor. Now, with the activation of this activity, there is cross phosphorylation of the receptor. So one partial unit of the receptor phosphorylates the tyrosine residues of the other unit and vice versa. Okay, so both of them cross phosphorylate, that is known as cross phosphorylation. This leads to further activation of the tyrosine kinase, and ultimately it will cause the phosphorylation of small G protein. This is not the same heterotrimeric G protein. This is other small G proteins are there, like RAB. Okay, so some other G proteins are there which are important as second messengers. Okay, so with the phosphorylation of small G proteins, these are activated, and this leads to further downstream events. So one protein leads to phosphorylation of other protein, leads to phosphorylation of other protein. So there is cascade of events. In fact, you see that everywhere we are talking about the cascade of events. One after the other, something happens, and this cascade of events is responsible for the amplification of the response. Okay, so with this, there is activation of MAP kinase. So I told you in the beginning of the video that there is activation of MAP kinase pathway. MAP kinase moves to the nucleus and causes the phosphorylation of the transcription factor, right? So with the phosphorylation of the transcription factor, this transcription factor becomes active, now binds to the DNA, and will lead to increase or decrease in the synthesis of certain proteins. Okay, so that is how there is this receptor tyrosine kinase pathway which is acting by the activation of MAP kinase pathway, causing increase or decrease in the synthesis of proteins. Okay, simple, you remember the name itself is receptor tyrosine kinase. So receptor is having this kinase ability, so phosphorylation of certain proteins is going to take place. Okay, the next pathway, that is the JAK-STAT pathway, where there is extrinsic enzyme activity. So similar mechanism is there. Here receptor is shown, and as the ligand binds to this receptor, one ligand which acts by this mechanism is leptin. Okay, so as the ligand binds to this receptor, there is receptor dimerization, and you see there is a protein JAK, that is Janus kinase, which is associated with this receptor. So with the receptor dimerization, there is JAK activation. Okay, and because of this JAK activation, now this JAK cross-phosphorylates each other. Okay, so one side receptor cross-phosphorylates the JAK on the other unit of the receptor. So that leads to JAK phosphorylation, that is known as cross-phosphorylation, or some books give it as autophosphorylation. So JAK phosphorylation occurs. When that happens, there is another protein, STAT, which is a cytoplasmic transcription factor. Okay, so this STAT goes and associates with the JAK. Okay, so there will be binding of STAT with the JAK, and this will in turn lead to phosphorylation of the STAT, right? So when STAT is phosphorylated, so it is not only one molecule which will be phosphorylated. Again, there will be cascade of events. There will be amplification of the response, and many STAT molecules will be phosphorylated, and then there will be STAT dimerization. Dimerization means two units of a STAT will come and bind together. So you see here, that is a STAT dimerization. So this activated transcription factor now enters into the nucleus and goes and binds to the DNA, thus increasing or decreasing the enzyme activity, right? So that is the JAK-STAT pathway. Again, very simple. Phosphorylation of JAK leading to phosphorylation of STAT, STAT dimerization, which enters into the nucleus and activates the transcription of proteins. Okay. So that was all about the cell membrane receptors. We saw ion channel linked receptors, G protein coupled receptors, and the enzyme linked receptors. In G protein coupled receptors, we saw the various second messenger systems. In enzyme linked receptors, we again saw various types of enzyme linked receptors. Now moving on to the cytoplasmic and nuclear receptors. In very brief, we are going to see, because the fundamentals we already know. So here it is shown the cytoplasmic receptor, and this red one is the nuclear receptor. So the steroid hormones, the thyroid hormone, the vitamin A, vitamin D, they are all lipid soluble, right? So these ligands can just diffuse through the cell membrane and enter into the cell. Once they enter into the cell, the steroid hormones bind to their receptor, that is the cytoplasmic receptor, steroid hormones. Okay. After binding with the receptor, there is again dimerization. Okay. So there will be another receptor here. Okay. And the ligand will bind to that, and then these two will combine together. They will move to the nucleus and bind directly to the hormone responsive elements in the nucleus, in the DNA. Okay, so there is increase or decrease in the synthesis of proteins. On the other hand, the thyroid hormones, vitamin D, vitamin A, they all enter to the nucleus. So they can diffuse through this membrane, they will also diffuse through this membrane. Okay, steroid hormones also diffuse, but their receptors are present in the cytoplasm, so they bind there. Thyroid hormone, vitamin D, vitamin A, they bind to the receptors which are present in the nucleus. They also form dimer. Okay, so there will be another receptor like that, and the ligand is going to bind to the receptor. They will form the dimer, and they will go and bind to the hormone responsive elements present on the DNA, leading to increase or decrease in the synthesis of proteins. Only one thing you remember here, that there can be formation of heterodimers also. Okay, that means one type of receptor associates with other type of receptor, like mostly they associate with the retinoic acid receptor, the vitamin A receptor. Okay, so that is how the cytoplasmic and nuclear receptors bring about the action. Now coming to that, how the response is stopped. So there are four fundamental mechanisms by which the response can be stopped. One, directly remove the ligand. Okay, so obviously when the ligand is removed, now the receptor will be inactivated, and that will not lead to further cascade events. Second, bring the receptor to its initial state. For example, we have seen in G protein coupled receptor that G alpha after activation binds to GTP. So this G alpha has an intrinsic GTPase activity. Okay, so once this GTP binds, automatically this GTPase activity of the G alpha is activated, and in small time this GTP is converted to GDP. So this brings about the heterotrimeric proteins in their initial state. So then G-alpha dissociates from the enzyme to which it is attached and then goes back and attaches to G-beta gamma. Okay, so again it forms this trimer. So that is second mechanism. And also there are mechanisms by which this intrinsic GTPase activity can be increased also. So there are regulatory proteins to increase or decrease this intrinsic GTPase activity depending on how the hormone is acting or how multiple hormones are acting simultaneously, right? Then third is inactivate or remove the second messengers itself. Okay, so inactivate like phosphodiesterase causes the inactivation of CAMP to 5-AMP. So now no more second messenger is present, so it is not going to act further. This we saw in CGMP as well. Remove second messenger, for example, the removal of the calcium ions, right? So we have seen that how calcium calmodulin pathway acts. When we remove the calcium ions, say suppose by calcium ATPases, so this is the membrane, there are calcium ATPases there, then we have sodium calcium exchanger as well. Okay, then in the sarcoplasmic reticulum or endoplasmic reticulum we have the smooth endoplasmic reticulum calcium ATPases, so they can also remove the calcium and bring back the calcium in the store. Okay, so once the calcium is removed, the response will be stopped. Then dephosphorylate the proteins, because we saw that ultimately most of the action is brought about by phosphorylation of the proteins. So there are phosphatases which are activated with time, which we saw in the beginning of the video, that is known as phosphate timer. So there is dephosphorylation of the proteins as well. So these are the various mechanisms by which the response is stopped in the cell. Now, before we go to the applied aspects, one fundamental you remember here, that though we have seen so many mechanisms, the response depends on the cell, which are the receptors it is having, how these receptors they are activated, and especially how the cell machinery is working. So different cells will have different proteins which are activated or deactivated. For instance, I will give you one example. Say suppose insulin. Okay, so same insulin acts on muscle, adipose tissue and also on liver, right? But everywhere it is activating different enzymes and everywhere it is causing different actions. Okay, so the mechanism of action by the tyrosine receptor itself, but with the activation different proteins are activated in different tissues and this leads to different actions in the tissues. Then sometimes same ligand can have different receptors. For example, we have epinephrine and norepinephrine. We have two different types of receptors. There are alpha receptors and the beta receptors. Okay, so beta receptors, they act mainly by CAMP mechanism, while alpha receptors act mainly by the phospholipase C mechanism. Then how many number of receptors are there, that also depends, because depending on the concentration of the hormones, the number of receptors goes up and down. That is known as upregulation and downregulation of the receptors. Okay, so this is some fundamental which you should remember. Now moving on to the certain applied aspects of this system. So we have seen so many receptors, and we have seen that the action of these hormones are brought about by binding to the receptor, forming hormone receptor complex. Now there can be receptor mutations. So there can be loss of function mutation where the receptor will not respond. Hormone is binding but the receptor is not responding. For example, in case of nephrogenic diabetes insipidus, there is mutation of the V2 receptor for ADH. Okay, so ADH concentration is okay, but it is not able to act on the receptors. Second is the gain of function mutation. In gain of function mutation, the receptor becomes oversensitive. Okay, so for example, in case calcium sensing receptor in the parathyroid hormone becomes oversensitive, so with the calcium concentration being there, it decreases the release of the parathyroid hormone too much. Okay, so because it is oversensitive, so we have hypoparathyroidism, but that is not because of the problem in the release itself, but because of the gain of function mutation of calcium sensing receptor. So that is known as pseudo hypoparathyroidism. Then we have various toxins also which act via these receptors. For example, we have the cholera toxin, which inhibits the GTPase activity for the GS type of GPCR. So with the inhibition of the GTPase activity, what will happen? The GS will become more active, right? Because as I told you that GTPase activity causes the breakdown of GTP to GDP, so as to stop the response, but that is not going to happen. So the action will be too much. This causes increase in the secretion in the intestine. Then we have pertussin toxin, which inhibits the GI activity. Okay, so that means that receptor now will not be able to inhibit the adenyl cyclase system. Okay, then we have various drugs which act via these receptors. In fact, G protein coupled receptors is the most investigated drug targets. Okay, for example, we have beta blockers, alpha blockers, we have histamine receptor blockers. So many receptor blockers are there which are targeting G protein coupled receptors. Okay. Then we have other mutations as well of this G alpha, G I, where for this itself, G alpha, G I, we have the gain of function mutation and loss of function mutation. And with time, we are finding more and more these kind of mutations being associated with different diseases. So that was all about the fundamentals of mechanism of hormone action. Thanks for watching the video. If you liked it, do press the like button, share the video with your friends, and do subscribe to the channel Physiology Open. Thank you.